Welcome to this video on ketone bodies, types, synthesis and utilization. These are alternative energy fuels generated by the liver under prolonged fasting, starvation and medical conditions such as diabetes mellitus. In this video we will explore their chemical natures, definition, enzymes and steps in their synthesis and utilization. Ketone bodies are ketonic compound represented by three. First one is acetoacetate. This is primary ketone body. This is used to drive the other two. Second is acetone, and third one is beta-hydroxybutyrate. Acetone and beta-hydroxybutyrate are secondary ketone bodies. These are generated by the liver as an alternative energy fuel under prolonged fasting, starvation, and certain medical conditions such as diabetes mellitus. During prolonged fasting and starvation, adipose tissues release fatty acids which are converted into ketone bodies. These ketone bodies provide energy to the brain and other tissue and reduce the need for gluconeogenesis from muscle protein. Therefore, production of ketone body is an adaptive response to spare muscle proteins during prolonged fasting and starvation. The major site of ketone body synthesis is the liver mitochondrion in prolonged fasting and starvation. However, there are reports indicating their synthesis in the brain astrocytes during ischemic condition. So let us discuss biosynthesis of ketone bodies, which is also called as ketogenesis. It completes in 5 steps utilizing 5 different enzymes. The first step is condensation of 2-acetyl-CoA molecule by thiolase forming acetoacetyl-CoA. Acetoacetyl-CoA further condenses with third acetyl-CoA molecule in presence of HMG-CoA synthase forming HMG-CoA. This enzyme is rate limiting regulatory enzyme, very important steps. So HMG-CoA subsequently converted into primary ketone body called acetoacetate. Acetoacetate is subsequently converted into beta-hydroxybutyrate secondary ketone body, which is the most abundant ketone body in our system, and this step is catalyzed by beta-hydroxybutyrate dehydrogenase enzyme. And finally, acetoacetate is decarboxylated into acetone. This is also secondary ketone body. This is volatile ketone body. Acetone can also be formed spontaneously from acetoacetate. We have seen that ketone bodies are produced utilizing acetyl-CoA, and for using ketone bodies as a source of energy, they must be converted back into acetyl-CoA. This is achieved in 3 steps and 3 enzymes. The first step is conversion of beta-hydroxybutyrate into acetoacetate by the enzyme dehydrogenase. This enzyme is also used in the synthesis of ketone bodies. Therefore, this step is reversible. Acetoacetate subsequently condenses with a succinyl-CoA, forming acetoacetyl-CoA with the help of thiophorase enzyme. This enzyme is unique. This is absent in the liver. Therefore, liver cannot utilize the ketone bodies as source of energy. Acetoacetyl-CoA subsequently forms 2-acetyl-CoA molecules with the help of thiolase enzyme. Thiolase is also utilized in the synthesis of ketone bodies, therefore this step is also reversible. Acetyl-CoA undergoes TCA cycle forming GTP and reducing potential, which after undergoing electron transport chain form massive amount of energy for cellular function. Ketone bodies are produced as alternative source of energy under the stressful conditions such as prolonged fasting or starvation. Therefore, they spare glucose for the renal medulla and RBCs. They ensure survival of other tissues under these stressful conditions, and also they serve as alternative source of energy for the brain, which primarily depends on glucose. Our brain can utilize ketone bodies efficiently. And lastly, these ketone bodies also spare muscle protein by reducing the dependence of gluconeogenesis on muscle protein hydrolysis. There are certain terms related to ketone bodies which must be clear to you without any confusion. The first one is ketogenesis, which refers to the synthesis of ketone bodies, and the second is metabolic state, which is known as ketosis. indicating higher level of ketone bodies. When the higher level is increased in the blood, this is known as ketonemia. When higher level is increased in the urine, that is known as ketonuria. And when higher level of ketone bodies is able to decrease the blood pH, that condition is known as ketoacidosis. So these four are the conditions, and this is the metabolic process of making ketone bodies. Thank you for watching this video. Please note that I will be posting videos every week on energy metabolism, including glycolysis, pentose phosphate pathway, gluconeogenesis, glycogenolysis, glycogenesis, TCA cycle, electron transport chain, and oxidative phosphorylation. So keep watching to update and refresh your biochemistry knowledge. Please like it, share, and subscribe it. Until next time, you take care. Bye bye.